Hey guys, so this week we're talking carbohydrates. Let's get into it. So carbs are our key molecule into energy production. So plants make carbs, i.e. sugars, and we consume them to burn those sugars, break those bonds, get the energy out. So that's really the cycle that's going on. Now, of course, you can substitute in other things. It doesn't have to be glucose, right? It doesn't have to be that. It doesn't have to be, it does have to be oxygen for the most part if you're going to do cellular respiration, but it doesn't necessarily have to be. It just usually is. If you've taken microbiology, you learn that oxygen is the terminal electron acceptor for many organisms. It's not the only one, but for the most part, you're going to use oxygen, you're going to burn some glucose, you're going to break some bonds, you're going to get some stuff out, but I have to make those bonds too. So we need to talk about carbs because they're an awesome, great form of energy storage. Now, that's not their only job. They also serve as an important structural component. And so we're going to talk about that. Let's talk about the actual name of carbohydrates. So the term carbo obviously means carbon, and then hydrates refers to the idea that if you look at the empirical formula for a carbohydrate, sometimes you'll see a little M here too. I can put it in here because that's not entirely true that it's that the number of carbons will match the N. Sometimes you can have extra carbons, but I left it off because, hey, why not? But you have carbon and then hydrogens and oxygens. Okay, that's your carbohydrates. So we kind of usually write the structural formula this way, and that lets us, that it looks like water. So hence hydration. Okay, that's why it's called carbohydrates. We're going to talk about all these things. We got monosaccharides and disaccharides, and oligosaccharides are not included here, and then polysaccharides. They're going to cover all kinds of cool things. So I'm going to use a piece of a video right now that shows just some of the basics, just going to get our brains thinking about carbohydrates. It's really, really good.
Carbohydrates include both simple sugars, which are little ring-shaped molecules made of carbon, hydrogen, and oxygen, either alone or in pairs, as well as more complex carbohydrates, which are formed when these rings link up together to make long chains. Carbohydrates provide us with calories, or energy, and simple sugars in particular play a lot of roles in our diet. They sweeten lemonade, balance out an acidic miso soup, fuel yeast in rising dough and alcohol, and help preserve jams and jellies. Now, sugars are found naturally in plants like fruits, vegetables, and grains, as well as animal products like milk and cheese. Added sugars are the sugars that get added to foods, like cereals, ketchup, energy bars, and even salad dressings. To be clear, even if the sugar being added comes from a natural source like sugarcane or honey, it's still considered an added sugar. In fact, a variety of ingredients listed on food labels might be sources of added sugars, some of which you're probably familiar with. Sugar actually refers to a family of molecules called saccharides. Monosaccharides, where mono means one, so one sugar molecule. Disaccharides, where di means two, so two sugar molecules linked together. Oligosaccharides, where oligo means a few, so it's 3 to 9 sugar molecules linked together. And polysaccharides, where poly means many, so it's 10 or more sugar molecules linked together.
This is from osmosis.org. They make really good videos. We're going to watch a portion of the rest of the video in a minute. So let's start talking a little bit, just some of the basics of our simpler carbohydrates. So our monosaccharides are what we're going to start with. But first we need to understand just the basic structures and how we're going to talk about carbohydrates, okay? So the smallest molecule that we can actually call a carbohydrate is called a triose. And OSE, we're going to encounter a bunch, so as a suffix. So OSE is a suffix. That's what we call carbohydrates, right? So galactose, fructose, glucose. So if it ends in OSE, it's a carbohydrate. Now, carbohydrates can fall into two different categories. They fall into either aldoses or ketoses, and that has to do with whether or not they have an aldehyde, which is their carbonyl on the end, or whether they are ketones, where they have their carbonyl group stuck somewhere in the middle and not on one of the terminal carbons, okay? And so that's how we organize these. Now, most of the carbohydrates that we're going to encounter are actually aldoses. Why is that? Well, it turns out when you have an aldehyde, so when you take that carbonyl and put it on the end here, it's easier to react with than if you have all these other things kind of blocking this double bond. And so you can just kind of physically imagine it's easier to get something into this space here to react with this bond to form something else. So like I could roll this guy, attach it to the other end of this molecule, which I'm going to show you in a little while. I could attach it to something else and just modify it, or I could come over here and cleave that bond and it would be easier. So aldehydes in general are more reactive than ketones because they have these extra carbons on the side are sort of protecting the reactivity of this carbonyl group here. Okay. Leads into the idea that comes up a lot with sugars and carbs and stuff is the ideas of chirality and enantiomerity or enantiomers. So a little organic chemistry in here, but it's not that bad. So what does it mean to be chiral? Well, that means practically it means that you have a carbon with 4 different types of groups attached to it. And so here we have our aldehydes. Okay, so it's one of these. Here's our D-glyceraldehyde. And here's the difference between D-glyceraldehyde and L-glyceraldehyde. Okay, we're going to talk about that in a second. And so with D, we have the hydroxyl group on this side. With the L, we have the hydroxyl group on this side. And because you have 4 different things, so you have this crazy looking thing here, you have a hydrogen, you have this group, and a hydroxide. So because there are 4 different things, that means I can sort of have a mirror image. Okay, so that's where our chirality, well, at least the chirality of our atom is concerned. Okay, and enantiomers is what we actually call the mirror images. So when we can flip these things over. Okay, right? If I put a line down the middle, you can flip these onto each other. So it's like your left hand and your right hand. They have all the same structures, but they're just in different locations, and so that's enantiomers. Now, stereoisomers, i.e., if you have chirality, basically all your saccharides, as long as they're aldoses, can engage in stereoisomeric forms. They can have chirality, and we're going to see some examples in just a second. And that can actually have a big effect on what they're used for. Glucose is probably the simplest one to actually look at. So here's glucose here. It's got 6 carbons, C6H12O6, awesome. But how we configure it, because we have chiral atoms, okay, we have chiral carbons, we have multiple chiral carbons, we are able to maintain the location of the R groups or whatever is attached to those carbons, okay? And so you can maintain those, and then as they're folded over or reactions happen, then they're going to maintain their location even though the content is the same. And so natural glucose looks like this. This is the D conformation. So you have 3 on the right and 1 on the left. That's kind of how I view it. Now, when you artificially generate glucose, okay, it's L-glucose, and all of them are on the left. See that? Okay. But we don't really care what the key one here is. We're looking at this chiral carbon here, okay? Because you can flip them. It doesn't really matter. This one is the chiral carbon that decides whether it's L or D, because that's what determines if it's glucose or not. I'm going to show you some later on. If I start flipping some of these hydroxyls, we actually change the name, okay? But you can see this isn't my favorite thing because it gets really complicated and some of the terms get really complicated, and so I apologize. But honestly, it's not too crazy. It's just wherever you put that hydroxyl group lets you change the name. It's not that complicated. So let's keep going. Let me just talk a little bit. I've been showing you Fischer projections, so that's when you linearize the carbons, that sort of carbon backbone that the sugars are going to be made of, okay? And if you fold those carbons over on themselves, okay, so if we actually form the bond here between, basically this oxygen is going to go attached to this carbon here, okay? And so if you imagine that you're sort of like on a plane here, so plane, so all the carbons here, so there's 1, 2, 3, 4, 5, 6 carbons. These 5 carbons here are sitting, existing on a plane, okay? And then off of that plane, your hydroxyl groups are either going to stick up, or they're going to stick up. This is actually supposed to be sticking down. So they'll stick up, the blue one's sticking up, and then these red ones are sticking down. So let me show you a better picture here. So we've got our carbons here. So there's our Fischer projection, okay, and they're colored. Okay, so there's the red one, the green one, the pink one, the blue one, other blue one, and there's the oxygen, okay. They actually form to this carbon here. Okay, and off of that, we can see where, so there's my plane of existence, right? So this is in 3 dimensions, and so one of the hydroxyls is sticking up, this one's sticking down, this one's sticking up, this one's sticking down, okay? Up, down, and if I change how those are arranged, it can actually change the functionality, it can change how reactive it is, it can change the actual name that we give them, which is kind of fun. Okay, so let's keep going. There's lots of different monosaccharides. It just kind of depends on how many carbons you're going to include. So you have your aldoses and ketoses, so that's your 3 carboners. Carboners, I just made that up. That's your 3 carbon molecules. And then as you add carbons, glyceraldehyde is an aldose. Okay, it's got an aldehyde on the end. Notice all of these are aldoses. Okay, and then you can also have a ketose, which is dihydroxyacetone is a ketone. Now, all the rest of these are aldoses, and so the number of carbons decides what number gets up front. So if I have 3 carbons, it's triose. If I have 5 carbons, it's pentose. If you have 6 carbons, it's hexose. Now, most of the, if you're not doing anything weird, then most of the time you're going to be encountering pentoses and mostly hexoses. But hey, sometimes if you're doing weird stuff, maybe you'll have a quadrose or whatever, tetrose. It doesn't even list them because that's like super rare unless you're doing weird chemistries. But we're going to assume that you're not doing weird chemistries. Now, of course, these are Fisher projections, so we can look at, what do you call these? Haworth projections, sorry, I forgot. So you can look at the Haworth projections, and so you can just see structurally what's going on. And I always center myself on the oxygen. So here's my typical glucose molecule looking right there, okay? Now, monosaccharides basically are, these 3 are the monosaccharides that we're going to be dealing with most of the time. Okay, you can have other monosaccharides, okay, so like riboses and deoxyriboses, those are pentoses, right? They only have 5 carbons, so we got 1, 2, 3, 4, 5 carbons, 1, 2, 3, 4, 5 carbons, right? That's what we use to make our nucleotides. When we're actually making our carbohydrates, like for energy, then we rely more on our hexoses, which have 6 carbons. So 1, 2, 3, 4, 5, and then you get that 6th one sticking off there. Now, for fructose, which is one of the monosaccharides, one of the 3, it's a little different. It kind of looks like the pentoses, but notice you got this extra carbon hanging off the bottom here. So sometimes people are like, well, those look the same. Well, they're not actually the same. It's just the carbons. I mentioned just a minute ago that where we put those hydroxyls, right, where whether on the top or the bottom, and I can change where these are on the actual molecule. This refers to, we can use those to keep track of the actual types of aldoses that we have, okay? And so you can see the D series, so these are naturally formed aldoses, okay? So aldoses just, they're aldehydes. This is most of the sugars that we're going to encounter. So you can see down here on the bottom, we've got our glucose, and we've got our mannose, and we've got our galactose, okay? Those are 3 biggies, okay? And so we've got our, we have 3 on the right, 1 on the left, 2 on the left, 2 on the right, galactose, 2 on the left, 2 on the right. And so you can just kind of see, basically, we just change the names based on where these things go. Arabinoses show up, riboses, there's your ribose, xylose, you'll see sometimes, but there's a bunch of these that just never show up, like gulose. I don't know, it's just really weird. Idose, they're just generally don't get made. You can synthesize them, but for the most part, but hypothetically they can exist. And so, yeah, that's what that is. One question gets brought up all the time. Who cares? Okay, well, it actually matters because, for example, if I'm going to build like a polymer of sugars, okay, so now we're not talking about polymers yet, but I just want to bring it up because it has to do with where hydroxyls are. So in alpha glucose, okay, so just to orient yourself, there's the oxygen, right? So we're looking at this first carbon here, and this first carbon is going to decide our that it's a glucose, or sorry, that this hydroxyl is down, so it's a glucose, and then we get to flip this one. Sorry, so the number 2 carbon decides if it's a glucose. The number 1 carbon gets to decide if it's alpha or beta. Okay, and so we're going to look here, and we're going to flip our hydroxyl down. That's an alpha. Okay, and if I link a bunch of those together with glycosidic linkages, which we're about to talk about, then I get this kind of formation of all these alphas. This is a typical formation for starch, which is a very digestible yet long-term storage molecule. Okay, so this would be your starches. So like things that you can eat, like I don't know, like complex carbohydrates. So when you hear complex carbohydrates, this is what they're referring to. There's these long chains of sugars. They're not monomers, they're polymers. And so that's what they form. But it's digestible. Now the key there is if I jump over here, okay, and I've got all these beta glucoses, so that's where the hydroxyl group is pointing up here, okay? And I'm going to kind of keep flipping them. So if I keep attaching all of these betas to each other, you can see they start getting like kind of this weird shape. So in order to get them to actually bond together properly, I have to flip them. So half of them are flipped. And so you see the little blue, the little yellow, blue, man, the little yellow hydroxyl groups that determine whether this is a glucose or not, pointing down, up, down, up, down, up, in comparison to each other, whereas these are all down. Derp, I'm just saying what's really obvious. If they're like that, that is cellulose. So that is an indigestible fiber, right? So if you're like, oh, I got to eat Wheaties because I need fiber or whatever, I don't know. You got to eat your leafy greens to get cellulose in your colon to help clear out your colon. That's what this is. So this is indigestible. So that one simple change, okay? So having that hydroxyl group either point down or point up can decide whether or not an enzyme is able to break it or not. Okay? Really interesting. So that leads us into these types of linkages, which these types of linkages let us form disaccharides. So that's where we're going to take two monosaccharides, okay, whatever they are, and stick them together using a glycosidic bond or a glycosidic linkage. It's just a dehydration reaction, okay? Or, yeah, confused. So there's condensation reactions and there's dehydration reactions. So dehydration reaction is the loss of water if during a condensation reaction, whereas a condensation reaction isn't necessarily a loss of water, it's just a loss of a small molecule when you're combining two things. So dehydrations are condensations, but not all condensation reactions are dehydrations. Whatever, you're forming a glycosidic linkage here between these two. Now I wanted to go back to that original video that we were watching, and they're going to talk about the different disaccharides that exist and the kind of foods that they're in.
Glucose is the most important member of the sugar family, and it's a monosaccharide. It's one of the main sources of calories for the body, and is able to cross the blood-brain barrier and nourish the brain. Another monosaccharide is fructose, which is commonly found in honey, fruits, and root vegetables. Finally, there's the monosaccharide galactose, known as milk sugar. It's known as milk sugar because it's only found in nature when it links with glucose to form lactose, a disaccharide found in the milk of mammals, which includes cow milk as well as human breast milk. Sucrose, or table sugar, is another disaccharide, and it's formed when fructose links up with glucose. Sucrose is found in various fruits and vegetables, with sugar cane and sugar beets having the highest quantities. Maltose is another disaccharide, and this one is two glucose molecules linked together, and it's found in molasses, which can be used as a substrate to ferment beer. Sugars, like fructose for example, are most always found in combination with other sugars, and the combinations can be pretty different, even in seemingly similar foods. For example, in honey, 50% of the sugar is fructose, 44% is glucose, 4% is galactose, and 2% is maltose. Whereas in maple syrup, less than 1% of the sugar is fructose, 3% is glucose, and 96% is sucrose. So simple sugars, whether they're natural or added, are mixtures of monosaccharides or disaccharides. Next, there are the complex carbohydrates. There are oligosaccharides, like galacto-oligosaccharides, which are short chains of galactose molecules, like those found in soybeans. Then there are polysaccharides, which are even larger chains with branches, and are the most abundant type of carbohydrates found in food. Starches are polysaccharides with molecular bonds between sugar molecules that human intestinal enzymes can break down. Starches are an important source of calories, and can be found in foods like rice, potatoes, wheat, and maize. Starches don't taste sweet like simple sugars, because they don't activate the taste buds in the same way. And there are also dietary fibers, which are carbohydrates that intestinal enzymes can't break down, and so the body can't digest them.
So just to make sure that, in case you're not watching the video, in case you're just looking at the slides, I just wanted to include this on here. The big ones, lactose you encounter in milk all the time. Sucrose is just your basic table sugar. And then maltose, we don't encounter like just purely that often. It's usually more of a mixture, but molasses is the big one that you encounter that in. And if you're making beer, you germinate the. I lived in Colorado where they make a lot of beer, so you get your malt and you germinate it in water, and then you get maltose out of it, out of the endospore, I guess. Anyway, doesn't really matter. It's just the different combinations of your various monosaccharides, and that's how you get your disaccharides. Awesome. Now let's move on to a little more complicated. We're going to start tacking on more and more sugars. And as we start tacking on more and more sugars, we call those oligosaccharides. So basically anything under 10 would be considered an oligosaccharide, though I'm sure you could find examples of oligosaccharides that exceed 9 sugars. But for the most part, most of our oligosaccharides stay under that boundary. Okay, so if it's 3, right, because it's a disaccharide, so the disaccharide is 2, so we're going to move on to trisaccharides and beyond. And that's basically, it's not really for energy consumption, though you could break these sugars off and use them for energy. They're more for signaling. So tons of signaling molecules are oligosaccharides by themselves or attached to proteins. And so just by themselves, they can be signaling molecules. So here are just a bunch of examples in human milk. Prebiotics, which is something, basically a prebiotic is what bacteria can consume that are probiotics that then like make your health better. So you can eat prebiotics to make the probiotics work better. It's a whole subfield that's mildly under my PhD, but I don't know, we don't have to talk about that. But anyway, prebiotics are interesting. You should eat healthy food is what this really means. Don't eat gross stuff because it's all artificial and all the sugars are artificial. You can see the variety. So I like these charts because they just, if you get super into this stuff, then you can just see like, oh, this one's got a fucose on the end. Oh, wow, that's very interesting. Ooh, and a galactose? Wow, that's very, very exciting. Anyway, you can see it gets kind of complicated. As you dig deeper, some people are super into oligosaccharides, some people are not. Doesn't really matter, but they do have a wide variety of associated functions. Okay, so from vitamin B to blood pressure changes to the absorption of calcium in menopausal women, like it's crazy. I clearly got this chart from an older paper because these are all from like when I was in high school. But just to give you some idea of the variety of functions that these really big oligosaccharides can have. Now, jumping off of that, they don't have to just exist by themselves. Okay, we can take oligosaccharides and attach them to proteins, specific protein residues, and we can then have them serve a function. So they could act as a cell surface receptor for something, or they can be a cell surface marker to mark a cell for whatever. There's so much variety that you can actually engender in these that it gets incredibly complicated really, 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 really fast. Okay, now there are 2 types. We call these glycans. So that's glycoproteins when we glycosylate. Okay, so glyco meaning like sugar, and sylation basically meaning we are attaching. So that's when we're attaching sugars to either lipids or proteins. Okay, and I'll show you some examples of lipids too. But for in this case, we've got like asparagine down here, right, which is our an amino acid. And so we're going to take some sugars and attach those. So we're going to take our oligosaccharide, our multi-sugar chain, and we're going to attach it to either an asparagine, okay, so that's called an N-glycan, okay, or we can have O-glycans, which that's where you attach it either to a serine or a threonine, okay. This is me being crazy. Crazy comment on cell surface proteins. Yes, you can have immune disorders where you are not properly either putting these on or you're putting the wrong ones on the cell, and that can cause an immune disorder because we use these to recognize cells for their functionality. Like, oh, is that a T helper cell or not? Well, that's a good question. Let's look at it. Let's look at their proteins and see if they have the correct oligosaccharides. And so it's really interesting. So they're added on in the rough endoplasmic reticulum because it's a protein, right? That's going to be put on the surface of a cell. So that has to go through the rough ER. And that's where you get your initial addition. And then it's going to go to the Golgi to have extra polysaccharides added. or extra saccharides, sorry. So there's a video that I want to show you. I apologize for the quality. It's like the best version of this video I can find, and it drives me crazy because I'm like, oh my gosh, this video is super good. The quality is like really bad, but the content is so good. So we're going to cut to that. It's going to show you this process actually.
Glycosylation, adding sugars to proteins, begins in the endoplasmic reticulum and continues in the Golgi apparatus. Let's examine the process for one protein. Enzymes in the ER remove glucose and mannose residues from the oligosaccharide chains. Vesicles transport the glycoprotein to the cis-Golgi network, where one of two events occurs. Either N-acetylglucosamines attach to the oligosaccharide and are transported to the lysosomes, or enzymes remove more mannose residues. Vesicles transport the modified glycoprotein to the medial Golgi cisternae, where N-acetylglucosamine attaches and more mannose molecules leave the molecule. Vesicles now transport the glycoprotein to the trans-Golgi cisternae. There, galactose and sialic acid join the chain. The glycoprotein is finally ready for transport to the cell.
Interesting stuff, right? I apologize for the quality again. But these videos from, they were really well done from some of the textbook companies, and then they just like never put the actual videos out. So you have to like find people who have like the CD from like the CD-ROM from like 20 years ago that have the videos still on it. Anyway, I also want to show you one other video. I'm only going to show you the first part. It's more to emphasize how complicated some of these can actually get, and that there is an entire sub-industry of biotechnology devoted to the addition or the analysis of these sugars on proteins. And so this is going to come from a pharmaceutical company that sells reagents to actually do this kind of work.
Secretory and membrane proteins are often post-translationally modified with sugar chains called glycans. Glycans are essential for the stability and function of the mature protein. They affect enzymatic activity, half-life, and receptor binding, among other biological processes. Proteins can be modified with different kinds of carbohydrates, N-glycans and O-glycans being the most common types in eukaryotic systems. N-glycans are attached to asparagine residues of the protein, shown with an N. N-glycans have a common biosynthesis pathway, which is reflected in the common structure of their core. N-glycans can be high mannose, which means all the component monosaccharides are mannose residues, with the exception of the two core N-acetylglucosamine or Glycnac residues. N-glycans can also be complex, which means the mannose residues are only present at the core and the branches are extended with other monosaccharides, such as N-acetylglucosamine or Glycnac, galactose, sialic acid, and fucose. O-glycans start with the addition of an N-acetylgalactosamine, or GalNac residue, to a serine, shown with an S, or threonine, shown with a T. O-glycans are often short chains consisting of only a few sugars. Or they can be extended by the addition of different monosaccharides, resulting in a longer chain. Deglycosylation refers to the removal of glycans from proteins. Enzymatic deglycosylation is the method of choice because it preserves the integrity of the protein and sometimes the glycan chains for further analysis.
Much higher quality video from New England Biolabs, or NEB as we usually call it, if you're a nerd. And they basically sell the reagents and the equipment to analyze the oligosaccharides on the surfaces of cells. And those are just ways that you can study a cell and see what it's trying to do. So where else do we see oligosaccharides? So here is an immune example. So up here, we have P-selectin and PSGL1. So it turns out that immune cells like to travel along your blood vessel. So this is the blood vessel. There's the blood vessel wall. There's your endothelial cells. Okay. and your immune cells. So here you got some neutrophils and some macrophages, or monocytes when they're flowing in the blood, but they're macrophages. And there's a T helper cell, and all these guys will express PSGL1 based on the actual presence of cytokines that are floating in the blood. Okay, and so if they're expressing that, they kind of reach out with little grabby arms and are looking for this molecule. So the leukocyte, or the neutrophil, T cell, whatever it is reaching out with this PSGL1, which is covered with all these sugars, and those sugars need to match up with P-selectin, which P-selectin is expressed on the endothelium. And so you can imagine, let's say down below where my cursor is down here. So down here, I actually have a, like somewhere there's damage. So maybe there's an invader, I've got a cut going on. And so you can see I filled the cut in with platelets here, but I'm going to express P-selectin nearby to reach out and grab the little arms of the PSGL1s from the immune cells. And so they only recognize those because they are glycosylated properly, which is really, really interesting. And bring it back to immunology. So glycosylation isn't relegated just to proteins, though. You can also glycosylate lipids. We call those glycolipids. Okay, if I take a lipid and I attach some kind of sugar to it, okay, those are glycolipids. There's a huge variety of these, just like there are glycoproteins. Just having them on the cell surface can signal things, but oftentimes you can also put them into the cell membrane. It can either add to stability, it can detract from membrane stability. It just kind of depends on what you're trying to do, and it has more to do with this end of the structures where we're actually adding. So this is the only glycolipid on here, is this one. It's just to contrast it with these other guys. But you could put it on the surface here, and that could signal to another cell, because this is what would be facing out, right? And this would be facing in towards the actual phospholipid bilayer. And so you can attach different sugars here, and it acts as a signaling molecule. Now you could embed a protein and put sugars on it too, like that's what we're doing here. But this is different, so you can just attach it right onto the lipid, which is kind of fun. Now another common example, blood type gets brought up all the time, but we need to talk about it because blood typing actually is based on glycoproteins and glycosylation of proteins. And so if you're O like me, this is what the structure looks like. And I apologize for the quality. It's because it's like a 2007 picture. So this is the actual structure. So you've got your galactose, you've got your N-acetylglucosamine, which is basically a glucose derivative with a nitrogen attached, sorry. And then you've got your galactose, and then you've got your fucose, okay? And so you can see that that same structure is in the A antigen as well as the B antigen, okay? And so that is maintained, okay? And so the only real difference between A antigens is that I tack on another N-acetylglucosamine, or I'm sorry, an N-acetylgalactosamine, oh man, or I replace that with a galactose. And you can actually express both of these, okay? So if you are AB, that means your cells are codominant. They are expressing both of these. So you would have, if you looked at one of your red blood cells, it would have an A antigen sticking on there, and you could look at other ones and they would have a B antigen on there, and that's the only difference, is this little square and this little circle, okay? But they all have the O possibility, okay? So that's why O is the universal donor, because it can go into any of these cells and they won't attack O as being foreign because they all contain the O antigen. So the O is just kind of like subtly hiding underneath the A antigen and the B antigen. So this is why you can't take an A antigen and put it into an O patient, okay? A blood and O blood don't mix, but an A patient can take O blood, okay? And so that's where that comes from. Hey, I brought it back to the immune system again. Now let's get to some of the fun stuff. Let's talk some polysaccharides. So if you have 10 or more, obviously that's a caveat. So it's basically more 10 or more repeating units. So that's contrasted with, I mean, obviously some of these structures can have more than 9. But for the most part, what we're saying is, okay, we have this continuous repeating unit. That's your polysaccharide. So yes, they are polymers, but yeah, they're polysaccharides. And so there's lots of different examples. Cellulose being like the indigestible component of plants. It's like your fiber when you eat fiber. And then there's lots of other things. So different kinds of starches that you can eat. And glycogen is what we use to store energy inside of our livers. Okay, we take glucose and we convert it into glycogen, and it's just a longer term storage molecule. You're basically, it's more efficient because you can cram more of it into a smaller space. and it lasts longer. It doesn't get digested as rapidly, and so you can store it for longer. Anyway, branching is important. So how branched it is controls sort of how accessible it is, which is interesting. And the actual structure of cellulose is really, really interesting. And so how they are organized, how we actually form those glycosidic bonds is really, really important. Okay, so there we have our beta glucoses where we're flipping them. Okay, and then the starches are the alphas. Glycogen and starch, notice, are really not that different. And then chitin, okay, is where you're using an N-acetylglucosamine in almost the exact same structure as cellulose. So you get that structurality. That's not the right word. The power of the structure, because you have this bond all going up and down. So you get the structural power of it. But it's just a different sugar molecule because we took an N and stuck an N on there instead of a hydroxyl group. But it's still, the structure is very, very, very similar. Okay? So that's polymers. Other polymers. So if you've taken a microbiology class, you may know that peptidoglycan is a really important component of basically all bacterial cell walls. And so you can see it here in the brown and green alternating. So that's the glycan portion. The peptido portion is the little amino acids that are connecting them together here. Okay, so gram-positive bacteria have a huge, massive, thick chunk of peptidoglycan. Gram-negatives have a nice thin layer. Mycobacteria also do have peptidoglycan. It's not as easy to kill them, though, because they have this giant, massive set of glycolipids. Hey, look at that. So we've got our lipid portion, and then we're going to stick on I guess this is the lipid portion, and then you got the little glucose or whatever it is. I don't actually know, which is on glycolic acid. But anyway, so you can see you're able to glycosylate, and that's really, really important for structure. And over here on funguses, which also have chitin as a structural component, there's other things too. So you've got proteins with a bunch of mannoses on them. You've got beta-glucans. So you can see it's not just for energy storage, but for structure as well. A couple things I wanted to talk about. I want to give you some actual examples. So because let's be honest, I struggle with keeping carbs interesting. I mean, I've tried to give you some examples where like signaling molecules or whatever, like you can dig really deep. But a couple examples that come out of things that I'm interested in. So vaccines I've always been interested in. And so there's actually a polysaccharide vaccine that we have. There's actually two types of them, and they're for strep pneumonia. So the trick with strep pneumonia, which causes like so many diseases, it's really bad. So it's streptococcus, right? So it's a little string of cocci, little circles. And they have a really powerful capsule. Powerful meaning it's like a really strong virulence factor. So that capsule of all these mucousy, sugary looking things prevents immune cells from attacking it. And so there's the bacteria itself, right? It's a circle, right? It's a little sphere. And because it's a coccus, and then it's got all these crazy, there's its cell wall, and then it's got all this extra capsular material, and it's tons and tons of different polysaccharides. There are 91 different types of polysaccharides just in the strep pneumonia capsule. That's crazy. There's so much going on. Okay, and so it causes a ton of human disease. Like it's really, really bad. It kills people. It hurts people a lot. And so what they tried to do was they said, okay, it's the actual proteins on the surface of the bacteria are hiding underneath the capsule. So can we make a vaccine against the capsule? Well, that's a good question. It gets a little tricky. So the trick with making vaccines that work against polysaccharide antigens is that sugar antigens, okay? So an antigen is any foreign molecule, right? And so you can imagine we've seen lots of examples of sugars. The actual, like, I have a mannose with a galactose and an N-glucosamine or whatever. The actual structure of sugars is not as different between creatures. Okay? And so it can be kind of tricky to actually make an antibody response against a polysaccharide. But you can do it. But the problem is most of the antigens that we deal with as creatures, as humans, as eukaryotes that have advanced immune systems like this, mammals, I should say. Most of the antigens that we deal with are proteins. And so most of the recognition machinery, so your MHCs, are designed to look for protein antigens. And so you can make antibodies against sugars. It's just more difficult to actually get the body to do it. And so that's why we conjugate them. So we take a pneumococcal conjugate vaccine. So that's the PCV13. This is one you want to get. You can get the pneumococcal, it's the non-conjugate, it's just the polysaccharide vaccine. That's this one. This will only give you passive immunity, so you kind of make some antibodies, but it's not great. And so what they did was, because the polysaccharides themselves from this capsule are not super immunogenic, they are different enough so we can attack them without attacking the body, but it's really hard to get an immune response. So what they did was they took the diphtheria toxin, okay, and it's a protein, it's the toxin that gets secreted by diphtheria, which is a bacteria, and it secretes a toxin and it can hurt you really bad. And so we take a little portion of that, we take the toxin portion off, obviously, and then attach that to the actual polysaccharide. And so that's kind of what's going on here. So here is the diphtheria toxin, okay, that's the diphtheria toxin. And so we took off the receptor binding domain. Okay, so where is it? Hang on. It's just a subcomponent of the actual diphtheria toxin, which has two components. The A portion is the one that will actually kill you. They disable ribosomes, which we haven't really talked about that much, but they stop ribosomes from being able to make proteins, so hence why diphtheria toxin can kill you. But we just take a little piece of that. It's super immunogenic, and so when you take just this little protein portion and stick it onto anything, that other thing that you attach it to, so it could be a polysaccharide or anything else, can become really immunogenic, and you can make antibodies against it. And so that's what this conjugate vaccine is. So that's what the conjugation actually is, where you're conjugating it. And so that's what this part is. So you can have just a polysaccharide vaccine by itself, but it doesn't work super great. But when you actually do that, you get really good B cell response, which is awesome. Now, the last thing I wanted to show you is a video, and that's just going to end this video, is a video of HIV, okay, because that was my field for like 10 years. And HIV has a bunch of glycoproteins, and so I wanted to show you a really good, well-animated video of the glycoproteins and how HIV uses its glycoproteins that it stole from a host cell somewhere else to actually invade another host.
HIV-1 attachment inhibitor. Proposed mechanism of action. Infection of a host cell by HIV leads to formation of new viral particles, death of the infected cells, and ultimately destruction of the host immune system. HIV particles carry GP120 proteins on their outer envelope, which play a vital role in allowing the virus to bind to host CD4 positive T cells. Attachment to the host cell represents the first step in viral entry and infection of the cell. This initial interaction between the virus and the cell, mediated by the viral GP120 protein, represents a novel target for antiretroviral drug development. HIV first binds to the cellular CD4 receptor present on the surface of the host cell. CD4 binding is mediated by the HIV GP120 surface glycoprotein, which together with the transmembrane glycoprotein GP41 forms GP160, which is present as a trimer complex on the viral envelope. In order to enable CD4 binding, GP120 must undergo a series of internal structural rearrangements. This process begins with rearrangement of the so-called inner and outer domains of HIV GP120. GP120 then undergoes further internal rearrangements in the region surrounding the CD4 binding site, which enable it to adopt a CD4 binding competent state. Once in the CD4 binding competent state, GP120 can bind to the CD4 cell. Following binding to CD4, GP120 undergoes further folding, which leads to exposure of its co-receptor binding site. Exposure of the GP120 co-receptor binding site enables engagement of the chemokine CCR5 or CXCR4 co-receptors. Following co-receptor binding, additional structural alterations expose GP41, which inserts into the host cell membrane. This initiates a series of major structural changes in GP41 that bring the viral and host cell membranes together, resulting in membrane fusion and release of the viral core into the host cell cytoplasm. This is where viral replication starts. The conformational changes that GP120 undergoes are critical in order for the virus to bind to and enter the CD4 cell. Attachment inhibitors represent a novel class of antiretroviral drug. They prevent the initial interaction between the virus and host cell, blocking subsequent viral entry into the cell. The attachment inhibitor binds to the outer domain of HIV-1 GP120 on the viral envelope, just underneath the region for CD4 binding. Once the attachment inhibitor is bound to GP120, the region surrounding the CD4 binding site can no longer rearrange into a CD4 binding competent state. As GP120 is not able to bind to the CD4 cell, subsequent viral entry is blocked. In summary, attachment inhibitors prevent the first step of viral cell entry, attachment to the CD4 cell membrane. This is in contrast to current entry inhibitors, which act after initial binding of the virus to CD4.